Okay, so today we are returning to our regularly scheduled content. We're going to finish up by talking about one final concept. We're going to be talking about the general anatomy of the central nervous system. So if you who have been out in lab already, which is most of you, have seen some of our central nervous system. We're talking about this first little bit, because in our next unit, we will be talking more about divisions of the nervous system. So reminders before we get going. So on Friday, when I see you next, we're going to just review. So there won't be a quiz for this weekend so that you have time to study, but a practice exam will go up. So keep an eye out for that. After our exam on Monday, the biology department has an in-work, like, what's it called? Biology classes are canceled on Wednesday, is what I'm telling you. So we will not have class on Wednesday. And check in with your other bio instructors if they have not mentioned that to you. We have a full day of meetings, but if you have a class at weird hours, I don't know. Okay, so questions about this. The exam will have the same structure as the last one, so that's what you're preparing for. Those quizzes that we do on the weekends are designed to sort of gear you up for this. And I'll put up a practice exam that's so that you know those practice exams are pretty directly paired to the question breakdown of the exam. Not the exact same questions, but like if I ask you a question about, I don't know, like a positive ion on the practice exam, that probably means maybe you'll see a negative ion on the actual exam, something like that, concept by concept. Not the exact same thing, but if you study the topics that are on the practice exam, that should be the same general topics on the actual exam. So I'm not trying to like trick you, and I'm trying to actually practice. Okay, cool. So our last topic is an intro to the nervous system. Okay, so nervous system can be kind of complicated until it clicks in your mind, at which point it will become easy. Here is a map of it. So you can see why it starts out complicated. But what we're beginning with today is just the central nervous system. So we're just thinking about the brain and spinal cord, where we have information processing going on. So that's our central nervous system. Has signals coming in from the periphery, and it's going to have commands going out through the periphery as well. So when we talk about this later, we'll see afferent coming into the central nervous system, efferent, E-like effect going out of the central nervous system. And we'll spend a lot of time down in here. So down in the breakdown of our efferent nervous system, the signals coming out of the brain and spinal cord, we're going to spend a lot of time comparing somatic versus autonomic, and then comparing parasympathetic versus sympathetic. Right now, we're just talking central and a little bit about peripheral, which is more of a, there are both functional and anatomical distinctions here. Central is because it's literally in the center of the body, peripheral because it's literally out towards the edges, and they have different functions in terms of information processing, integration, versus sending and receiving signals. Okay. So in our central nervous system, our brain and spinal cord, we have neurons and we have glial cells. We're going to be thinking mostly about the glial cells that are in the central nervous system, but we will touch base on a couple that we see out in the peripheral nervous system as well. So the neurons are what we've been talking about so far. Those are our excitable cells, excitable because we can change the membrane potential, send an action potential down the neuron, send a signal, right? So they conduct impulses. They generally don't divide. You may have heard, like, you lose a neuron, it's not coming back. There are a couple places in recent research we've seen that you can make some new neurons. But usually the traditional way of thinking about neurons is that they make new connections to other neurons, but we don't generally see more neurons, especially in the central nervous system. But cool topic of ongoing research. The glial cells aren't going to be sending action potentials. They're not excitable cells, but they are part of our nervous system because they support these neurons. They're going to coat them. They can divide. They protect these neurons. They're going to have roles in protecting the nervous system, supporting it both physically and physiologically. So we're going to run through some of the major functions of those support cells today. So when we think about the central nervous system, which is what we're thinking about first, CNS is the brain and spinal cord. It's necessary for maintenance of homeostasis. When we talked through those little workflows of homeostasis, those little loops, right? We saw that we had integrating centers where we're planning a response and then sending that signal out, right? So that's the role of the central nervous system, really, is to be receiving a signal, planning the response, and then sending all the instructions to create that response. There are 10 to the 11th neurons, so like 100 billion neurons with 100 trillion synapses. So we talked about one single or maybe 2 or 3 synapses at a time last week and on Monday, but there are 100 trillion of them. So notice that is a lot more synapses than we have neurons. So that's going to remind us that those neurons are connecting to many other neurons. Okay. So that central nervous system, it's responsible for basically consciousness, right? So everything you perceive, everything you do, everything you feel, everything you think, that is an effect of your central nervous system firing off these action potentials. It's really crazy. It is the least understood of our organ system. So as we were doing that activity on Monday, you may have hit a point where you're like, I can't find any answer to how this works. There is still lots and lots of research into how the central nervous system works, how the brain works, especially. Yeah. In terms of the general anatomy, right, we're going to have, we're going to have our neurons, which we've focused on so far. Today, we're focusing on our glial cells. We would also have physical support. So that's going to be your skull, you've looked at in lab, the meninges, which you either have looked at in lab or will look at in lab, as well as cerebrospinal fluid. Then we're going to also need blood supply for the central nervous system. So the central nervous system needs a lot of oxygen. We're going to need ATP to maintain that resting membrane potential. We're also going to need glucose to produce that ATP. There's also a special feature of the central nervous system, something called the blood-brain barrier. So in the blood out in the rest of your body, there's a lot of movement between the contents of the plasma and your tissue. But the central nervous system has extra layers of protection. We don't want everything that's in your blood getting into your brain. And there are some special features here that allow us to separate out those contents. We're also going to think about gray matter versus white matter, which is basically going to mean like where we have cell bodies, where we have anything unmyelinated, it's going to be gray matter. Where we have white matter, myelin is white. So that's mainly what we're talking about when we talk about white matter. Okay. So a neuron, physically, is the structural and functional unit of the nervous system. We've been looking at how it receives signals. We've been looking at how it fires off action potentials and secretes neurotransmitters. All right, so its role is to respond to chemical stimuli, so other neurotransmitters. Also, neurons can respond to physical stimuli, especially when we go out and talk about like sensors out in muscles, for example, nerves and neurons that we see out in muscles. There are some receptors that like literally respond to like something getting stretched, right? So that's the kind of thing we might mean by a physical stimulus. So far, we're thinking about chemical stimuli because we've been thinking about neurotransmitters. The neuron is going to conduct an electrochemical impulse down that axon, release our chemical regulators, aka our neurotransmitters, and ultimately enable our perception. So most of them can't divide, but they can repair. So we're not, hopefully, completely losing them all the time. Neurons can be classified based on what they do or what they look like. So based on function or structure, or function. So in terms of structure, we look at the neuron, we look at its shape, and we look at how many connections it has and where. So number of processes extending from the cell body. So that means we're looking at like where those dendrites are, where those axons are. Ooh, are they? Okay, I'll pop into yours then. Oh, okay, I'll pop into yours. Yours are these? Yeah. Did you get yours from Brightspace? Yeah. And those are what you. Okay, let's look what's in there right now. Did you download yours really earlier? Maybe it's, yeah. Okay. I mean, I didn't change them like super recently, but I did delete some stuff at one point. So maybe you got them beforehand. I'll go into the ones you have. Okay, let's see. Basically, I shortened some stuff for you at some point so that you would have a little less on the exam. All right, let's see here. So is this what you have? Okay, cool. Let's do this. All right, all that stuff that I was saying to you is true. But I think that means I cut some content for your exam so you'd have less to study. Okay, shortened it. Okay, so we have neurons and supporting cells. The supporting cells are the glial cells. The CNS is the central nervous system. Sorry, we won't have the red text, so I'm gonna have to guess what I meant to put there. So we need these for homeostasis. We've got lots of them. So neurons conduct impulses, can't really divide. Glial cells support the neurons. They don't conduct impulses, but we can make more of them. Okay. So this is still what I was talking about before. This is classification of neurons. Has a picture. I think your full slides probably have all of these. It's just you have additional ones with double information. For anyone who got these earlier than others. Okay, so when we think about the functional classification of the neurons, we're going to be basing it on the direction that we are sending these signals. Okay, so the sensory neurons are going to be conducting impulses from a sensory receptor to the central nervous system. Okay, so our sensory receptor, like say a receptor out in your finger senses that you touch something, right? This will then conduct an impulse into our central nervous system. So this is a picture of spinal cord here as our example. Spinal cord, that's just what we're looking at. Okay, so that's a sensory neuron. And later on, we will call these afferent neurons. Okay, you may or may not have seen this in phys lab yet, but afferent. Okay, so afferent neurons coming into the central nervous system. Okay. Motor neurons are how we get things going out. So motor neurons, both of these we're going to count as motor neurons. This one and this one. So these bottom two. So all that is about the direction of the signal flow. Right, so we're taking something from the central nervous system and going out. Sorry. So these are the efferent ones that make the effects. Efferent. So those are the motor neurons sending out signals to target organs. So when we think about target organs, we're usually going to be thinking about effector organs, we'll call them. So the ones we're going to focus on most in our next unit are going to be these ones, our smooth muscle, cardiac muscle, and glands. But when we get into the muscles, we'll also talk about sending signals to skeletal muscle. A neuron that's located just within the central nervous system can be something called an association neuron or an interneuron. So if you have connections in your brain that say just in your brain, conducting impulses like from the right hemisphere to the left hemisphere, something like that, this would have a different classification because it's not going into the central nervous system, but it's also not going out. Okay. All right, So next are glial cells. So the supportive structure of the brain is going to be composed of neuroglia. And I can also just post these with the red text afterwards. Okay, so our supportive cells are going to be composed of neuroglia. This is still part of our nervous system. It's protecting the nervous system. So here are astrocytes. Their most important function is going to be something called the blood-brain barrier. Okay, so astrocytes forming the blood-brain barrier. We'll see an astrocyte. All right, so here we see the astrocyte. Okay. All right. Our ependymal cells are going to line cavities. So your brain has cavities filled with cerebrospinal fluid in structures called ventricles. So here we see one here. So our ependymal cells are going to help us make that CSF, among other things. So they're lining these cavities. Our microglia are basically like the immune system of our central nervous system. So they're going to protect the central nervous system from foreign matter through phagocytosis. Okay, so basically they're eating pathogens. Okay, so they're going to be pulling them in and breaking them up. And then we're going to have two different types of glial cells that produce myelin. And so this is where we think about central nervous system versus peripheral nervous system. So in the central nervous system, we're going to have our oligodendrocytes. And in our peripheral nervous system, we're going to have something called Schwann cells. Okay. Both of these create that myelin. So remember, the myelin was where we have a neuron, okay, an axon terminal. And if we have an unmyelinated neuron, we have to trigger an action potential here, then here, then here, all the way down each patch of the membrane. But if we have myelin wrapping around it, so I'm going to put some myelin in blue here. All right. I'm going to keep the pink what we have to trigger, right? So now we only have to trigger an action potential in the gaps, right? So this is what speeds up conduction because we only have to trigger an action potential in these gaps called nodes of Grandier. Okay, so the oligodendrocytes and the Schwann cells are important because they're making that myelin, speeding up our conduction. So glial cells are often one of the things that are breaking down in some neurodegenerative diseases. So it's not always that you like physically have neurons dying off. It's often these supportive cells that are dying off or creating scarring or somehow malfunctioning. So they can't protect your neurons in the way they usually would. So this is the case in things like multiple sclerosis, right? So multiple sclerosis is a loss of myelin. So actually losing that protective coating that usually speeds up conduction, that gets messed up. Suddenly the conduction gets a little wonky, right? So those places where there used to be myelin and now it's missing, scar over, and this has a lot of effects on your function. Alzheimer's disease also involves plaque or scar tissue surrounding neurons, as well as loss of some specific types of neurons called cholinergic neurons. We'll learn about cholinergic later, but cholinergic refers to acetylcholine, which is one of our neurotransmitters. Parkinson's disease is associated with inflammation coming from the glial cells, right? So they're inflaming and causing basically like swelling. We're also losing neurons that produce dopamine in Parkinson's. And these are, of course, generalizations. Disease states have a lot of different possible causes and a lot of different things going into them. But these are three that are nervous system diseases. So physical and structural support for the central nervous system, we have the skull and the meninges, right? So the skull and the meninges we've looked at in lab. One thing that I wanted to highlight for us here is the cerebrospinal fluid, because looking at a liquid in a preserved body is difficult. So that cerebrospinal fluid is located in the ventricles, these holes inside the brain. So it's a clear watery fluid that's created inside the brain and then wraps around and bathes the brain. So this is secreted by those ependymal cells. one of our types of microglia, so glial cells, in a specific region called the choroid plexus, right? So it's secreted by these cells inside the ventricles. And then that fluid circulates through the ventricles around the brain and ends up reabsorbed into the venous blood flow through something called arachnoid villi, which are basically like release valves in the arachnoid matter, which is one of the layers of our meninges. So those of you who have not had lab yet and have lab on Thursday, you'll see that in that structure. Okay. So the function of the CSF is partially for like physical support and cushioning, but this also helps maintain a stable interstitial fluid environment, right? So we've talked about the fact that interstitial fluid, so the fluid outside of cells versus the contents of the cytoplasm, the fluid inside the cells, that intracellular fluid, can be different. And we've seen that our nervous system, these neurons, right, allowing sodium to rush in during that action potential, allowing our membrane potential to spike to positive 30. That is dependent on the electrochemical driving force, right, which we talked about in our last unit and focused in on a little bit about that setup for our neurons on the first day or so of class here. So when we're thinking about the CSF, right, because those forces are dependent on concentrations, among other things, and on those electrical gradients as well, we want to make sure that the fluid surrounding these cells is stable and has that same content all the time, because if it doesn't, suddenly we don't have that force for sodium to drive in, as an example, which means suddenly our action potentials aren't going to happen the same way, and we would have real problems. So it's a lot of explanation just to kind of explain what that means by a stable interstitial fluid environment, right? We're maintaining these gradients so that we can maintain these driving forces so that conduction always works the way it works. Right? So if we suddenly have a lot of extra salt in the brain or something, that would change how an action potential works. You don't need to memorize these, but these are some examples of compositions that differ between plasma, so the extracellular fluid in your blood, versus the cerebrospinal fluid, this fluid surrounding the brain, right? So we're going to have a little less glucose in the cerebrospinal fluid. We're going to have way fewer proteins in the cerebrospinal fluid. You can see that some of these ions are similar, but we do have some differences here in cerebrospinal fluid versus in the plasma. That does matter. So now we're going to talk about that blood-brain barrier, because that's part of why this can happen. Okay. So the central nervous system accounts for about 2% of total body weight. So usually it's about 3 to 4 pounds, okay, in life. But it actually receives 15% of blood supply. So it is getting a lot of blood relative to how much space it takes up in your body. Okay. So this second bullet point will be brain. Okay, probably. Okay. The brain needs glucose and oxygen. Okay. So we have so much blood flow because the brain is going to need a lot of glucose and a lot of oxygen, even at rest. So when you're resting, just sitting around, not even doing anything hard, you're going to be using 20% of your body's oxygen and 50%, like literally half, of the glucose in your body for making ATP, right? So this is because the brain really needs to be using glucose specifically for energy, right? So we need this blood flow going to the central nervous system. So this is why in lab, we also hammer on how we get that blood there. So neurons specifically depend on aerobic glycolysis. And aerobic cellular respiration in general, right? So when we talked about metabolism, we talked about aerobic cellular respiration, where we have oxygen, right, get all the way to the electron transport chain and oxidative phosphorylation. versus the kind of glycolysis that we can do anaerobically without oxygen, where we form lactate from the pyruvate and just kind of cycle through in the cytoplasm. Your neuron really needs to go all the way through this process, needs oxygen, and therefore it needs this steady supply of glucose and it needs a steady supply of oxygen. Super limited ability in your nervous system to use anything other than glucose for energy, right? If you are literally starving and dying, maybe you can use some ketones, right? But you really need glucose in order to get your nervous system, your neurons firing appropriately, getting the ATP they need to fire off those action potentials, okay, and maintain that resting membrane potential between your action potentials, like specifically. So if you decrease blood supply to the central nervous system, and specifically to the brain, that is a huge problem because now you don't have oxygen and now you don't have glucose. And so now those neurons are not going to work. Okay. So a stroke is just a decrease in blood supply to nervous tissue, or to brain tissue. So when we have a stroke, that can be really one of two things. So we'll talk eventually a little bit when we talk about the cardiovascular system about like ischemic stroke versus hemorrhagic stroke, or maybe you've heard those in your real lives. But basically, you can have a decrease in blood supply for one of two reasons, right? So either you can block a blood vessel. Occlusion, to occlude means to block, right? So like you're thinking like a blood clot essentially, if you have occlusion of a cerebral blood vessel, right? So that's one cause of stroke. I think that's a more common reason that we think about when we think about people having stroke. But the other thing that can happen is that if you have some type of damage, like hemorrhage, right? If a blood vessel in your brain gets cut somehow, right? Or you just have something burst, right? Bursting. This can cause hemorrhage. And if you are losing blood, it's not getting to where it should be. So this also is a type of stroke. So the blood-brain barrier is going to be made up of capillaries and our astrocytes, essentially. So the astrocytes were those glial cells responsible for the blood-brain barrier. Okay. So here we see a typical capillary. So capillaries are made up of something called endothelial cells, which you'll see when we talk about the cardiovascular system in detail, right? And they have some gaps, right? So they have some gaps between these cells called pores, right? So here we see pores. And this is where hydrophilic solutes, so anything that dissolves in water, can potentially cross between the plasma and our interstitial fluid, right? We don't want this to happen in the brain, right? We really, really want to control what is going to be in the plasma versus what is in that CSF, what is in this fluid surrounding the brain. So we block it. So we can do two things. So these green things are the astrocytes, right? So we can see them covering over some of these sections, right? So the astrocytes are partially responsible here for pushing these cells together, kind of pushing these endothelial cells closer together. It is also true that we have different types of cell junctions here. So we specifically have something called a tight junction between the cells. Tight junctions basically glue one membrane to another membrane, press them up right against each other so that we don't have these pores and things can't get through between the CSF and that plasma anymore. So the only thing that can cross the blood-brain barrier is something that we allow across, right? So things that can go through carriers. Carriers are proteins that we produce, put in those cell membranes to carry specific things across, right? So they can get through if we let them. Or hydrophobic solutes. So we talked about small non-polar things being able to cross their membranes. So specifically what we're thinking here is that like oxygen and CO2 can cross across these cells because oxygen and CO2 are non-polar, can dissolve across membranes. So we're not blocking them by pressing these cells together. So this is a special kind of type of anatomy in the capillaries in the central nervous system that limit the amount of exchange, right? So that's why we see a much lower level of proteins in the CSF than we do in the plasma, right? Because proteins are big, they don't even really fit through carrier proteins. and they can't get through pores, right? So they're not going to end up in the CSF. If they do, that's a sign there's a problem. So next we're talking about myelin. So the myelin sheath here, the myelin sheath in the peripheral nervous system, right, is going to be made up of our Schwann cells. So that's probably what's here. Schwann cells. Okay. So our Schwann cells are going to be wrapping around and around and around and around and around an axon, forming something called a neurolemma or neurolema, right? So that's what we're looking at here in this image. So this blue part is the neuron, right? And specifically the axon. You can see one little cell body here. And then this stuff is the myelin, and it's made up of just wrapping that Schwann cell around and around and around. So this is how we're forming the myelin sheath in the peripheral nervous system. So like a nerve out in your arm, the myelin is going to be made up of these Schwann cells. It's going to look like this. So we do still have gaps. So when we talked about that saltatory conduction, that's the fancy word for that jumping conduction of action potentials, right? Here, 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 right? Those are the gaps between the Schwann cells. So here's another picture, just an actual picture of what this looks like. So these are the Schwann cells wrapping around, forming the neurolemma, right? Forming this myelin sheath. So this is a myelinated axon. If you have a really small axon, it's going to usually be unmyelinated, so it's not going to look white, it's going to look gray. Even the unmyelinated axons in the peripheral nervous system, we might think of as having a neurolemma because they don't wrap so many times around, right? Like the wrapping over and over of these Schwann cells is what's making the myelin. I think this is probably supposed to say that this is called a neurolemma. All right. So the myelin in the peripheral nervous system, we just said is made by those Schwann cells. Okay. And this is a summary side about myelin, right? So our Schwann cells are wrapping around the axon, forming the myelin sheath, which we call the neurolemma if we're in the peripheral nervous system. But in our central nervous system, these are going to be made by oligodendrocytes. Oligo, it's just this word from the next bullet point. Dendrocytes. So the oligodendrocytes don't work exactly the same way as the Schwann cells. They still produce myelin, but if we look at an oligodendrocyte, you're going to see one cell body here, and then you can see it has multiple projections. So it has like these feet, and it's grabbing onto multiple different neurons, right? So it's grabbing onto multiple different axons. So this is a different structure in the central nervous system than we saw in the peripheral nervous system, where we just have a single Schwann cell wrapping around and around. So our myelinated axons have this white color. Myelinated axons. Okay. So on this image with the oligodendrocytes, our gray matter is here in this darker beige, this kind of brown color, versus white matter is in this lighter beige. So what you can see is that in the gray matter, largely what we see are our cell bodies, right? Our soma of the neuron and our dendrites, right? So receiving the signals here in the gray matter, our cell bodies, and then out as we're projecting out with these axons, if we're myelinated, we have white matter. So we're going to physically see this in our nervous system. It will literally physically look white versus gray. Okay? So when you see gray matter, you got cell bodies. When you see white stuff, most of our nerves out in the peripheral nervous system are myelinated, so they're going to look lighter in color. That indicates that there's myelin. So the nodes of Ranvier are those gaps between the myelin, which we circled on the Schwann cell side, right? So there, right? The gray matter is in the cell bodies and in the dendrites. So we're really going to see our gray matter almost entirely being within the central nervous system because of that structure where we're signaling from the central nervous system out to the periphery, right? The cell bodies, that gray matter is really going to be located, the cell body within the brain or within the spinal cord, and then it's going to have like a long projection out into the body. Okay. So you do also see some white matter in the central nervous system. It's that you don't really see gray matter in the peripheral nervous system. Okay. Yeah, I did super shorten it. That is all we need to know about glial cells for our purposes for the exam. Okay, so you are free to go, but I'm going to stick around and ask questions. Well, yeah, and you can ask questions. And then on Friday, we'll be reviewing. Yeah, your test is on Monday.